Hello children, once again I am back with another branch of biology, not so new, but it has contributed a lot to the field of medicine, agriculture and industry, and that is biotechnology. And we are having two chapters under this branch, biotechnology, that is chapter 11 and chapter 12. Chapter 11 will deal with the principles and procedures, and in chapter 12 we will be dealing with the applications of biotechnology. So, let us get started. So, first of all, let us define biotechnology. Biotechnology deals with the techniques of using live organisms or enzymes from organisms to produce products and processes useful to humans. So, once again I am reiterating the definition. It deals with the techniques of using living organisms, mostly microorganisms or enzymes from organisms to produce products and processes useful to humans. So, in that context we can say making of curd, wine, bread, which we do in the household, is biotechnology because it is microbe mediated process. So, all these are microbe mediated process. So, biotechnology can range from these simple processes of curd making, wine, bread making, etc., which we do in our household, and it is carried over centuries, this technique. So, that is biotechnology. It ranges from here till the most sophisticated technique like IVF, gene therapy, making of vaccines, DNA vaccines, rDNA vaccines, genetically modified crops, genetically modified organisms, etc. So, it ranges from the simple making of curd, wine, bread, which are done in household, to the very sophisticated techniques like IVF, which we have dealt in chapter 4. Gene therapy, again we will be dealing in chapter 12. Then making of rDNA vaccines, and now you know the coming vaccine, the vaccine that is going to be available in the market and much awaited vaccine is the COVID vaccine. It is going to be produced by rDNA technology, and we have already learned hepatitis B vaccine is a product of rDNA technology. Also GMCs, genetically modified crops or transgenic crops, genetically modified organisms or transgenic animals, all are included under biotechnology. So, once again this gives an overview of what is this branch of biotechnology. It is not so new, but a branch that has a lot of scope and contributing to the various fields and bringing about success in various areas like medicine, agriculture, industries. So, once again reiterating what is biotechnology. It is a technique that deals with the techniques of using living organisms or enzymes from organisms to produce products and processes useful to human beings. And I told you what and all comes under biotechnology, right from the simple making of curd, wine, bread, which we do in our household, to the very sophisticated techniques which we will be dealing in chapter 12. Already we have dealt with IVF. It includes IVF, gene therapy, then vaccine production, production of genetically modified crops, production of genetically modified organisms, all that are for human need. It is not for the benefit of the organism, but for our need, to satisfy our needs. So, when we deal with these topics, GMCs and GMOs, we will understand what cruelty is done to these animals. Now, actually these two scientists, Herbert Boyer and Stanley Cohen, they were the first to construct an rDNA. And this construction of the rDNA was in 1972, and this laid the foundation stone to biotechnological industry. So, the discovery of the rDNA or the construction of rDNA by these two scientists, Herbert Boyer and Stanley Cohen, the contribution of these two scientists, their contribution was that they constructed an rDNA, and this laid the foundation to the biotechnology industries. So, this was the main basis of biotechnology, the construction of rDNA, and they accomplished this task in 1972. As we proceed, we will be dealing with the construction of an rDNA. So, remember these two scientists in 1972, they were the first to construct an rDNA. Now, what is an rDNA? rDNA, if I give a diagrammatic representation, rDNA can be represented like this way, that is this circle. I will make a complete circle. So, you can see circles or circular DNA in prokaryotes. What are they called? Plasmids. Okay, so this is the plasmid. Now, into this plasmid, a gene of interest which I am giving yellow, which will stand out, is incorporated. So, this yellow segment indicates gene of interest. Now, as I proceed with biotechnology, I will use terms like gene of interest or desired gene or alien gene or foreign gene. Preferably, I will go with gene of interest, which I have given an abbreviation GOI, which is not standardized. So, this representation, you can see a white ring, that is the plasmid of the host. It is a plasmid of the host. So, we know the host is definitely a bacterium, and bacterium is represented, or E. coli is represented by an oval shaped structure. E. coli or any bacterium. Okay. So, that is the plasmid, the white ring, and you can see on the white ring I have given a patch of yellow which shows the gene of interest. Okay. This representation, what I can give it in a formula, host plasmid DNA plus gene of interest is what is called rDNA. This should be in your mind. What is an rDNA? Host plasmid DNA. That is, the green E. coli bacterium is representing the host, and the host plasmid is white in color. That host plasmid DNA along with the gene of interest that is incorporated constitute the rDNA. How can we make this rDNA? Now, we are going to see how is this rDNA constructed. And we know this was accomplished by two scientists, Herbert and Boyer. So, how it was constructed? Now, BT is correlated with IT. Okay, when IT had a boom in the 2000 millennium, at the same time BT also gained popularity. So, IT and BT are always correlated, and IT use the term in the quick access toolbar we have cut, copy, paste, and you are very familiar, it goes like that in the protocol, cut, copy, paste. It is cut, then copy, paste. It always go in this order, whereas in BT, so this is about IT, you can correlate with BT. BT means biotechnology, it is again cut, starting is cut, but then it is paste and then only copy. So, you remember these techniques, that is the protocol for IT is cut, copy, paste, and that is at the tip of your tongue, cut, copy, paste, but here it is cut, then paste and then only copy. So, there are tools that are required for cutting, there are tools that are required for pasting or gluing, and there is a tool for copying or cloning. Okay, we better word in biotechnology we use cloning, or we can use copying. Okay. So, now I gave it in brief for you to understand and correlate, so you will never forget. Okay. So, now we are concerned with the construction of rDNA, and I told you what is rDNA, plasmid DNA plus foreign DNA. So, genetic engineering in a nutshell can be called genetic modification. Another name for genetic engineering, genetic modification or genetic manipulation, where we directly manipulate the organism's genes using biotechnology. And biotechnology, there are very important tools. We will be coming on the next slide, we will be dealing with the different tools handled in biotechnology, but I am just giving the tools used. There is a, as of now I will say it as molecular scissors, there is a molecular glue, then there is a vector as well as the host. And from the molecular scissors is meant for cutting, molecular glue is meant for pasting, and the vector host is only involved for copying, as I told, cut, paste, copy, but vector is needed. Vector plays the role of a vehicle to transfer the gene of interest. I will come to that. As of now, just we are dealing superficially and we can go into the depth later. So, genetic engineering is called genetic manipulation or genetic modification, where we are directly manipulating the genome or DNA of the host organism using biotechnological tools. Also, to make the concept clear, it involves various steps, and coincidentally the step starts with alphabet I, the verb starts with alphabet I. So, remember there are 4 I's involved in the steps of rDNA technology. Once again, the 4 I's are: first we have to identify the gene of interest. Again, you will understand this only when I deal with the construction of an rDNA in detail. As of now, remember these verbs. First, we will identify what? The gene of interest. Then next is we are isolating that DNA which has the gene of interest. Then third step, we are inserting the gene of interest into the plasmid, and finally we are introducing the gene of the rDNA into the host bacterium. So, first of all we are identifying the gene of interest. So, I will just show it schematically. I took the color yellow for gene of interest. So, let the DNA which had the gene of interest be yellow. So, all of you focus at this DNA. I drew a helix representing the DNA, and we are first the entire strand of DNA. Looking at the entire strand of DNA, we have to first identify where is the gene of interest. So, first important step is identification of the gene of interest. So, just imagine we have identified, let this be our gene of interest. Okay. We have identified our gene of interest. For example, to go about with an example or substantiation, I will take the case of insulin. So, let this segment which I have highlighted in red be the gene that produces the protein insulin, and you know the role of this insulin. Okay. It is a hormone which is involved in bringing down the sugar in the blood. So, it is used for the treatment of diabetes. So, I am going with an example which we all are aware. So, let this be the gene that expresses. So, we have learnt in the previous chapter, expression means transcription, and after transcription, translation. Translation, we get a protein, and here the protein of interest is insulin protein. It is produced by this gene which is highlighted now in white. So, there are a number of genes in this DNA, but we are not concerned with other genes. So, I will be indicate other genes. This is another gene, this is again another gene, all with different functions. We only have to identify the desired gene. So, here we are concerned in insulin production. So, first step, we identified the gene of interest. So, that step, identification, is done. Next step is we have to isolate what? Should we isolate the gene of interest? No, we cannot isolate the gene of interest alone. We have to isolate the entire DNA. So, this entire DNA should be isolated. This full DNA strand should be isolated. So, that is the second step. Isolation of the entire DNA from the cell. Okay, this is inside the cell. It should be isolated, and this step we will be doing in the lab. Now, next step is we got the entire strand of DNA. So, I will pull this out and I will show you once again the yellow entire strand of DNA with our gene of interest. I will indicate our gene of interest with a red portion. Okay. Now, we are only concerned with the gene of interest. So, now I pulled out the entire DNA strand from the cell. Next step is to cut. We need only the gene of interest. So, we are using the scissors and cutting this part. Okay. After cutting, see, I got only one small segment. This is our gene of interest. So, I cut and I got this segment. We got only a small fragment, gene of interest. And I told you we make use of the molecular scissors to do the cutting. So, the first tool I used is scissors. I cut, I got that segment. That segment is the gene of interest. Here the gene of interest is the insulin gene. Next step is we have to incorporate this gene into the plasmid. This should be incorporated into the plasmid. So, actually the plasmid should be white in color, complete white ring, okay, host plasmid. Now, this yellow strip should be inserted here, okay. So, I am placing it here. I am talking in context with this template. Actually, it is not pasting over the plasmid. The plasmid itself should be cut. So, before that we have to cut the plasmid. So, the plasmid is cut. So, in that gap, so if I erase that portion, this is the plasmid, and I told you this plasmid is cut. So, this gap. So, here also cutting is involved with the help of the molecular scissors. Now, in this gap, this yellow strip is inserted. So, inserted. Once inserted, we have to put glue. Now, we got the rDNA. So, we got what is rDNA? I told you host plasmid DNA plus the gene of interest. Now, I got the gene of interest incorporated into the plasmid, but this is all out of the E. coli. It was not inside. So, very importantly, I must remove that. It was all done outside the host. Now, I must put it inside the host. So, the last step, it is introduced into the host. Finally, it is introduced into the host. It is not as easy as this. Just simply I am drawing a line, an oval around this. Not simple as that. There is a step by which this rDNA gets into the host. But to introduce these terms, I just gave you a briefing. So, first we identified the gene of interest from the entire DNA. So, first identification. Next, what we did, we isolated the entire DNA from the cell. So, this entire DNA was isolated. So, it is taken out. I isolated it. Next step, which is not there, it is understood we have to cut that segment. See, that segment is cut. See, that cut segment is here. This cut segment should be inserted into the plasmid. See, it is inserted into the plasmid, into the vector or the plasmid. The usual vector is the plasmid, and finally, what is it? We have to introduce it into the host bacterium. So, as we proceed, we will understand these steps very clearly. As of now, remember the 4 I's. Here the 4 I's indicate. The first I is the verb identification, second is isolation, third is insertion, and fourth is introduction. But it should be in complete statement. Identification of what? Identification of the gene of interest on the DNA. Second is what? Isolation. What is to be isolated? Not the gene of interest alone. The entire DNA should be isolated. This step you will get familiarized with this step because we are going to have a lab on it. So, this you will never forget, the entire strand should be isolated. Third step, what we are doing, only the gene of interest, this is the step, only the gene of interest is inserted into the plasmid. That is the third step. And fourth step is we are introducing this plasmid. Now, this plasmid is called an rDNA. This plasmid is introduced into the host, and the host, usual host is bacterium. Clear? Once again, this will be clear only when we move to the steps and go in advanced stages. Okay. As of now, remember these verbs: identification, isolation, insertion, introduction. Later, when I get back to this slide, the statements now will be very clear. So, what is rDNA technology? rDNA technology can otherwise be called gene manipulation or gene modification, where the genome is manipulated. See, I manipulated, and it involves 4 I's: identification of the gene of interest, isolation of the DNA from the cell, third, inserting the gene of interest into where? Into the plasmid, and finally introducing that plasmid now, which is called rDNA, into the host. So, this again I am telling you, it is incomplete. we didn't involve to concentrate on the eyes given in the textbook, we didn't concentrate on the cutting. This will be complete only after we proceed with the third and fourth slides. So, moving to the next slide, the tools. We are going into the tools of genetic engineering. Okay? So, the next slide is dealing with the tools of genetic engineering. The first tool is the molecular scissors, and here the molecular scissors is the restriction enzyme. We will have a separate video on this topic, molecular scissors, where I will be telling there are the different types of restriction enzymes. Anyway, I will be introducing the two now itself, because in DNA fingerprinting as well as in HGP, I already told you about which restriction enzyme is used popularly. So, restriction enzymes, there are two types: restriction endonuclease and restriction exonuclease. Anyway, the purpose of both these enzymes, or a restriction enzyme in general, is for cutting the DNA at specific sites. Very, very important, the keyword specific site. Not anywhere we can cut, only at specific sites. So, that is the first tool. Second tool is the molecular glue ligase. Ligase, the purpose is for sticking the DNA. Okay. So, earlier I showed you about the plasmid host DNA with the scissors, molecular scissors, we cut here and here. So, this part is vacant. Okay. And into this, what did we do? The gene of interest was inserted. Now, what we have to ensure it is glued. So, let this blue color be ligase, glues here and here. Now, we get what is the product after gluing? We get an rDNA. So, as I told you, first cutting, copy paste, here it is first cutting, then pasting, and then only copying. But before copying, this rDNA, as I told you previously, this rDNA by itself outside, there is no use. It should be introduced into the host. So, that step is very important. It should be introduced into its host. Okay. Then only it will be complete form, and within the host it multiplies. Now, going by the tools, the third tool is the vector, and by now you know which is the vector. The vector is a plasmid, or it can be a bacteriophage. So, here I wish to define a plasmid. We have learnt in 11th standard, plasmids are small circular DNA found in prokaryotes in addition to the chromosomal DNA. So, if I draw a prokaryotic cell, okay, here there is already a prokaryotic cell, that is bacterium. Here we have the chromosomal DNA of the bacterium, that is chromosome, and in addition to this there are small circles in the cytoplasm. These circles are called plasmids. So, we have already, I have a picture of the structure of what is a plasmid, circular DNA. Okay. Now, what is a bacteriophage? Bacteriophage are viruses invading bacterium, and we have learnt in Hershey and Chase's experiment. It is a virus. So, I am just drawing the virus which is represented there. So, let this be the virus. This virus which invades what a bacterium is known as bacteriophage. There are three types of viruses: the virus that invades plants, virus that invades animals, and the virus that invades bacterium. When they invade bacterium, what viruses they are called? They are called bacteriophage. So, the vectors can be either plasmid or bacteriophage, and the purpose is to transfer the gene. See, when it got incorporated, see, the gene of interest is this. When it got incorporated into the plasmid, it is used, this plasmid is used as a vehicle. See, this plasmid or this is used as a vector. The purpose of the plasmid, it serves as a vehicle or as a vector to transmit it into or to transfer it into the host. Like we have heard of vectors of certain diseases like malaria, the actual parasite is Plasmodium vivax, but it is transferred via the mosquito. So, when we get bitten by a mosquito, it is transferred. Just like that way, the vector plays an important role, and hence it gets the name vector, vehicle for gene transfer. And lastly, the fourth tool is the host E. coli. Preferred host in biotechnology is E. coli, but in 12th chapter we will be dealing with other than E. coli, there can be other hosts like the plant cell and animal cell, but usually all biotechnological experiments are done on E. coli. The reason of E. coli, by now you know, it multiplies quickly, and for one replication it requires just 18 minutes. So, we can get a number of E. coli. So, we can get a clone of E. coli. That is the advantage of transferring it into E. coli. Multiplication rate or reproduction rate is very high. So, the purpose is for cloning the rDNA. So, it is inserted into the host, the rDNA is inserted into the host, and once it gets into the host, what happens? The host multiplies. 2020 round figure, within days you can get, or within hours you can get millions of hosts. And when the millions of hosts multiply, what happens in our gene of interest also gets multiplied because they are present in the host. So, actually we can say this host acts as a factory. Another name for host, when we are calling this as molecular scissors, that is restriction enzyme as molecular scissors, ligase as molecular glue, plasmid as vector. See, we are going to see the typical genetic protocol that is followed in biotechnology with the help of this video. With the same example substantiated, we are going ahead. The video also deals with the production of insulin. Okay, so let's get started. That insulin that is produced by rDNA technology is called humulin R. We are going to see the various stages, how we get it. It is a result of genetic engineering. Bacterial cells serve as factories that produce the human insulin for the treatment of diabetes. This is the bacterium, and I told you the parts of the bacterium. It has the chromosomal DNA or bacterial chromosome as well as a circular DNA called the plasmid. Only one is shown now. There are many such plasmids in the bacterium. It is very, very important. It serves as a vector. Now, bacteria has plasmids, and what are these plasmids? I have told you, they are circular DNA. Now, the next tool is the bacterium itself. The bacteria itself serves as a next tool, that is the host, and the advantage is that it is unicellular and reproduce asexually in minutes. You get quick clones. Now, the first step from the E. coli bacterium. Now, here the host is E. coli bacterium, and the source organism is the human cell, where inside the nucleus we have the DNA, and the gene of interest is the insulin gene. So, the first step, we are isolating the plasmid from the E. coli. At the same time, we are isolating the DNA from the human cell, which has the gene of interest. Next step, using the restriction enzyme, we are cutting the plasmid. At the same time, the same restriction enzyme is used to cut the DNA. We get the gene of interest, that is insulin gene. And look at the cut ends. The cut ends, there is a specialty, it is called overhanging, single stranded and sticky. Remember these three words. The cut ends will be overhanging, single stranded and sticky. Now, what is that? It is inserted, and like a jigsaw puzzle, it correctly fits because it is overhanging, single stranded and sticky. Now, we make use of ligase, which will paste the cut ends, and now we get what is called the rDNA. See, we got the recombinant DNA, which has the host DNA indicated in blue and the gene of interest, insulin gene, indicated in red. Now, what is the next step? The last step is this rDNA should be introduced into the host E. coli. So, we have the last step. The rDNA, as I told you, the blue and the gene of interest should be inserted into the host. That process is called transformation, by which the rDNA gets incorporated into the host, and this host is placed in a culture medium, and what happens as a result? It multiplies and we get clones of cells, and when we get clones, what happens? Our gene of interest also multiplies. Now our concern is not the gene of interest, it is the protein of interest. So it is the protein of interest is made to express itself to give the insulin protein. So the gene of interest was the insulin gene, and we get the protein of interest, insulin, and which is finally extracted from the bacterium and bottled and is used for the treatment of diabetes. So the final stage is expression. So that video gave you a clear cut idea, and so getting back to the same slide, see, this step indicates isolation of DNA from the human cell. So let the human cell generally be called the source organism, and correspondingly we are isolating the plasmid from E. coli. So this is isolation of plasmid from E. coli. So the first step is indicated. Human cell, from the human cell what is taken? The entire DNA, and from the E. coli what is taken out? The plasmid. So first step is isolation. Then second step, before that we have identified the gene of interest. Okay, so we'll check out, here's the gene of interest. Assume this is the gene of interest. Now the third step is involving cutting. So using the restriction enzyme, it is cut, and here it is cut. So the same restriction enzyme is used to cut both the DNA as well as the plasmid. Now into the cut ends, the next diagram shows into the cut or vacant space what is inserted, our gene of interest is inserted, and with the help of the enzyme ligase pasted. So we get an rDNA. So this represents an rDNA, and finally this rDNA is incorporated into the host. The host is E. coli, and this process is called transformation. And don't forget, this is maintained in a culture medium on large scale or small scale. Small scale in petri dish, large scale in bioreactors. And what happens? The host multiplies and we get clones of the host, which means we get clones of the gene of interest. And we are not concerned with the gene. See, the gene is incorporated into this. It is final step is the gene is made to express. So we know what is expression. It is forming mRNA, and then it is translated into proteins. So finally this gene, which codes for insulin, is expressed into the proteins called insulin. That's what we saw in the video. Finally the gene of interest is being expressed into the protein of interest. So now with the video and with the schematic representation, you got an outline of the steps involved and the involvement of the different tools of biotechnology, mainly the restriction enzyme for cutting, the glue, that is ligase, then the vehicle or the vector, that is the plasmid, and finally the host, which serves as the factory. Last, I'll wrap up with the principles of biotechnology. The first principle is genetic engineering, and this we will be dealing in lesson 12. Okay. So what is the first principle? Altering of the DNA or RNA and introducing it into the host. It can be a plant or animal, and thus the phenotype of the host changes, and the final product will be a GMC or GMO. GMC means genetically modified crop, or GMO genetically modified organism. The second principle of biotechnology is chemical engineering. That is what we saw in the slide or in the video presentation. It is the maintenance of a transformed microbe. See, I told you the last step where we see the E. coli. I am representing E. coli like this, and its chromosomal DNA with an rDNA. So E. coli with the rDNA. rDNA is what? Host plasmid DNA plus the gene of interest, which I have indicated in yellow. Now what is this E. coli called? This is called a transformed E. coli. or transformed microbes. So, where final step I told you, it should be maintained in a culture medium. So, the second principle is maintenance of a transformed microbe. So, this is a transformed microbe. A non-transformed microbe also I will draw side by. Same structure, I will use the same colors. E. coli represented by an oval oblong structure in genetic engineering or biotechnology. Its own chromosomal DNA and plasmid DNA, and here look at the plasmid DNA, no insert. This is a non-transformant. Non-transformant, and this is a transformant. I hope you identified this is a transformant, this is a non-transformant. Non-transformant, the plasmid is intact, no manipulation, whereas in transformant, the plasmid is inserted with the gene of interest. So, first you must identify transformant and non-transformant. So, here we have the two pictures, non-transformant and transformant. Now, here maintenance of what? Are we maintaining this? No, we are not maintaining, we are maintaining this structure. That is the transformant, E. coli with the rDNA. We are maintaining in large quantities in culture medium for the manufacture of biotechnological products like antibiotics, vaccines, human recombinant insulin, as we saw, humulin, growth hormones, blood clotting factors, hepatitis B vaccines, and it goes on the list. So, the last step we saw now, maintenance of the transformed microbe where in culture medium on large scale, and I told you for large scale we need bioreactors on large scale, and to get what the gene of interest is finally expressing into protein of interest which are useful to man. There can be some antibiotics, it can be vaccines, it can be humulin, it can be growth hormone, it can be even clotting factors. And I have given the picture, see this is the picture of the insulin developed by rDNA technology, and that is called humulin. It is available in the market, it starts with humulin with another ending suffix or prefix, okay, but it is humulin, from that you can guess it is produced by rDNA technology. Then we have the hepatitis B vaccine, see this is the bottle or the ampule which has the hepatitis B vaccine, which is the product of rDNA technology. Then this is a blood clotting factor which are used for hemophiliacs. These are administered, so they are given, these hemophilic patients are given this, so at any time they have this injury they administer themselves with this. It is a blood clotting factor, and we are having a number of drugs or medicines that are produced by rDNA technology. It can be ranging from antibiotics to simple drugs, all by rDNA technology. So, this is the second principle which we will be learning in chapter 11. See here the second principle in chapter 11 and the first principle in chapter 12. So, once again reiterating the two principles of genetic or biotechnology. First principle is altering the genome and incorporating into them. Therefore, it produces a favorable genotype. Altering the DNA and introducing it into the host. Therefore, we get a favorable phenotype. The genotype is altered, we get a favorable phenotype. That is, this template is indicating the first principle which we are actually going to learn in chapter 12, and the second principle is this, the maintenance of the transformed microbe. I told you how it becomes a transformed microbe when it has an rDNA. When it is not manipulated, it is non-transformed. So, we are maintaining this transformed microbe in culture medium, and that too on large scale for the production of certain products like insulin, then vaccines, coagulating agents, drugs, etc. So, with this I wind up the first video which deals with the steps involved in rDNA technology and the principles behind rDNA technology or biotechnology, and the two persons who won the Nobel Prize in Science for their contribution to this field which has become a hallmark in the field of biotechnology. They are Herbert Boyer and Stanley Cohen. They accomplished this in 1972. Thank you.